This is an interesting way to start the biobank talks, with talking about what is going wrong with the biobanks. So I am an assistant professor at Columbia, biomedical informatics and computer science, and I am a core faculty member at New York Genome Center, and my research, as I mentioned downstairs, is generally translational bioinformatics, and I have this special interest in privacy and security. I'm not a cryptographer, but I use applied cryptography to make the analysis of omics and clinical data more secure. So today's talk is going to be about that, and how do you scale it for biobank scale data, basically. So I want to first start with something that everybody knows, that we live, it's actually so exciting to be in this field, because we are getting data from everywhere, right? We are getting data from genomics, epigenomics, transcriptomics, and we are getting data from electronic health records, some data from ICUs, high frequency data, and imaging data. And there are also other types of data like the mobile apps, and so much data about human health. It comes with a lot of analysis challenges that all of you are trying to address, but it also comes with these other challenges, such as how do we maintain the privacy of the patients who are giving their data. I guess it goes without saying that the patient data is private. I mainly work with two types of data, genomics, and this includes also all sorts of omics data, transcriptomics, epigenomics, and also electronic health records. And many of you know our genomes, we share 99% of our genome with each other, but that 1% difference makes us unique, but also it makes our genomes very unique, such that even in the identified form, it can be very identifying. And there are several papers that I'm not showing here that shows that the genomes cannot be anonymized. Electronic health records is a little bit a different story, because when it comes to genomes, we actually have policies and protections where, for example, insurance companies or employers cannot do discrimination based on genetic information, but insurance companies work with electronic health records. So it's a little bit of a different story, but it is extremely private, and because of that, for example, when you're pulling data from hospitals, we have several hoops that we have to jump through to be able to get access, and anonymizing health records, especially clinical notes, extremely difficult and perhaps not possible. But having said that, we cannot not share data. Especially during the pandemic, we learned that data sharing can save lives. It accelerates research, accelerates diagnosis, and in fact, the biggest discoveries have been made because we could share data. Like today, we talked about the Vanderbilt BioRxiv, which is an amazing resource where all those discoveries were able to be done because the researchers had access to that data. So then, how do we actually create systems where we allow researchers to do these type of analysis in a more responsible way, and that we can open up the data to many more researchers for them to collaborate? So I'm going to talk about how we can apply what we learn from applied cryptography, both two types of data, both how do we learn from genomes, and then the second, how do we do analysis with the clinical data, collaborative analysis with the clinical data. So the first one was actually inspired by this resource that we've been trying to build at the New York Genome Center, where we are trying to collect data from psychiatric patients all over New York. And these patients are very vulnerable. Most of them are coming from very different backgrounds, both ethnically and also economically. And the goal is to create this resource of basically genotype and phenotype data. And the reason for that is that, as was mentioned, that most of the biobanks that we have access right now, especially UK Biobank, usually contains a lot of healthy patients. So when you want to focus on a specific phenotype or disease, it's very difficult. It's less so in all of us that we have, because the recruitment was done in medical centers. But still, if we want to reach huge sample sizes for understanding the effect of rare variants, especially, we need to perhaps build these resources specific to a specific phenotype. And then the current system, ecosystem, if we look at UK Biobank and all of us, it's actually great, right? You have this cloud sandbox where you go as a researcher inside and you do your analysis in there, and on the hope that nothing is leaking, basically, no private information. But the world is not that simple. So there could be, for example, attacks to cloud servers where the data can be breached. There's always these possibilities. So then we ask the question, what if we build something where the data, it's still in the cloud, in the server, but it's encrypted and you can interact with the encrypted data set? Can we do that, and can we do it at scale? So I also want to mention, beyond biobanks, if we work with data that is deposited to NIH, for example, whole genome sequencing of psychiatric disorders, WGSPD data set, you can apply in dbGaP and you can get access to. It also sits in the cloud through Anvil, but let's say we are a researcher and we want to do some analysis. The traditional way of it is that you apply for access, you get access to data, you download it locally, and then you do your analysis. And the data sits actually in dbGaP in some sort of encryption. But the shortcomings is that if you have data from, I don't know, hundreds of thousands of patients, you have to have local resources. When you download the data, you have to have the data storage and GPUs, CPUs to be able to do the analysis you can do locally. And also, once the data is shared, the data owner cannot revoke the data access. It cannot remove any patient records, or it cannot guarantee any privacy protection, right? If it's at the hands of a student who doesn't know any better, data might leak. You might find it in a website, or it might leak through your code in GitHub. Maybe you have some MRNs or something about the patient. So there is no control in the system how the data is handled after the data is being shared. So then yes?
I want to ask a question about the local computation. Like, my intuition is as data grows, we will need to move the computations
to the cloud.
To the cloud.
Yes.
And as you said, that data leakage is very dangerous and security is the risk. So why will we need to increase the number of local computations?
No, so that's the idea, right? So you don't want to, so maybe you're not allowed to put the data to cloud in the unencrypted form. For example, in Europe, there is GDPR, and in certain cases, the personally identifying information, even sometimes pseudo-anonymized form, cannot be put on cloud. So that means that you have to do your compute in the local computers to provide security, right? And then now the idea is that if we have this very vulnerable data set and we don't have the local resources, can we actually create systems where we can put the data in the cloud and do the computation? So that's why we want to use in-use encryption for that. And we developed this database called Secure and Queryable Database. It's very telling from the name, but we call it SQUEET. Don't ask me what I stands for. You can ask my students. So we use in this database this technique called homomorphic encryption, which enables operations between ciphertexts. So you encrypt the data, and you can do operations on the encrypted data. But it's very limited. You can only do summations and additions and multiplications. And I'm going to very briefly, this is a very simplified math of how it works, just to give you an idea. So in homomorphic encryption, you have a message M. It's a public key cryptography. You encrypt your message with your public key. And then if you want to decrypt it, you have to have the secret key, which is the private key. And you basically express all of the data in polynomials so that you can do basically operations between them. And how it works is that if you have this polynomial M plus kx, you have two messages that you want to sum up. You create ciphertext 1 and ciphertext 2, and then this is basically showing that proving that if you add those ciphertexts and decrypt them, you will get what you would get when you add them in plain text. So
sorry.
Yeah.
K is the public key?
No, here I'm not showing even public key, But this function is not just a function of the M, but it's a function of the PK, which is the public key. And the K is, public key is a function of K. So K is basically a, this is the polynomial, this is a random variable. Your message, so this is some kind of noise that you're adding to your message. But you're adding that, sorry, go ahead.
Sorry, my ignorance, but X is the random variable.
Yeah.
So that's the noise, and K is something that is a function of your public key?
Yeah, yes. Your public key is function of K, basically.
What's the difference, because it's not invertible, or?
Oh, because then here you have your secret key, so it shouldn't be invertible, so that you can't decrypt it with your public key. You can only decrypt it with your secret key. And then, so when you decrypt it, you get the results in an so you get the results encrypted form, and now when you decrypt it, you get what you're supposed to get, basically, from the summation. And then the multiplication works in a similar way, basically. So basically, if you have two polynomials, you can multiply them and sum them. So if you express your data in the form of polynomials, you can do multiplications and summations on those polynomials, and then when you decrypt it, you will get the plain text results. So that's the very simple way of explaining, perhaps not exactly accurate way of explaining it, but just bear with me. Basically, the idea is that with homomorphic encryption, you can add and multiply. So basically, think of now, we are trying to create a biobank where we have tons of patients and we have their genomes, and now we have some kind of phenotypes. You can add as many columns as you want. You can also add things like smoking status and social determinants of health. And in this database, we turn them into categorical variables, basically. So we can handle floats, like the continuous variables as well, but it makes it a lot smaller because it's slower, because in homomorphic encryption, the kind of libraries that we use that are efficient usually work with integers. So everything turns into an integer space. So the idea is that, can we create a database, put it in the cloud, and then allow researchers to work with them, interact with them? So that, can we create queries that only uses additions and multiplications, or can we approximate some queries that uses additions and multiplications? But there are 3 challenges to do that. One is, so the homomorphic encryption were designed for 2 parties. So I'm the data owner. I encrypt my data, put it on the cloud, do some computation, and I take it, and then I decrypt it with my own secret key. So if I'm going to allow multiple researchers to communicate with that database, then I have to give them my secret key. But then what is the point, right? So how do we solve that? So that's the challenge one that we worked on. The other one is high storage cost. Basically, it's 100 times more expensive to store data, so we need to figure out a way of compressing the data, because the genetic data as it is already large. And then another one, as I mentioned, we can only use multiplications and additions, so then we approximate everything with multiplications and additions, and they are already slower than the plain text multiplications and additions, so it creates a huge overhead. So in this particular project, we try to basically address all these 3 challenges. First, we came up with an idea to overcome the 2-party challenge that was inspired by this thing called in cryptography, this thing called proxy re-encryption. We call it public key switching. So basically what we want to do when a researcher communicates with the data set and computes something, the results should be encrypted under that researcher's secret key, so that only that researcher can decrypt the results. So what happens is the data owner collects all the researchers' public key. So that's like you're applying to dbGaP to get data access. You send them your public key. Public key is public. They authorize you, and then they create a key switching key based on your public key and their public key. And they store that key switching key with the data set in the cloud. So when I interact with the data and do some computation. Everything is done under the owner's key, but then at the end, they take the results, and then they switch the key of that results to my public key so that I can decrypt it. And this is done also homomorphically so that there is no decryption of the data. It is re-encrypted without any decryption, so the server never ever sees the data or anyone's key.
But the data needs to be duplicated.
It's not duplicated, it's the results. So,
No, only the result.
yeah, only the result.
A version of the data that is encrypted with
The data, only the results is going under key switching. So everything is done, it's still on the owner's, under the owner's key. So only the results get switched and sent to the, and the results are not stored anyway.
But the user needs the owner's key?
No, the key switching allows to switch the key of the results, so that when they,
The key without re-encryption?
without decrypting, yes.
Just a clarification, what do you mean by what's the range of results?
I will get to that.
Results is a big object.
Yeah.
Sorry.
Yeah, I will get to that. It's usually a vector as with the number of patients that you have. Like if you have 100,000 patients, then it's a vector of 100,000. But I'll tell you, we have 4 functionalities, and it depends on the functionality. So that's how the key switching works, so that when someone queries, they get the results under their own key, but there is no re-encryption of the data itself, but just the results. And then the high storage cost is actually simple to solve, because in actually homomorphic encryption in certain libraries, there is this thing called batching. It's the single instruction multi-device SIMD batching, which takes a bunch of data points and then compresses into a single point, such that you can still do the queries and the computations on the single points that are batched together. And that actually reduces the data storage. This is not something we came up with. This already existed. And actually, I can show you how much gain you get. So this is the unpacked version, the first one, and then the second one is the packed version. And you see that you gain a lot from the data storage. However, it's still a lot larger than the plain text. And IS is it's an encryption method, a very common encryption method that is used to store data, but it doesn't allow you to compute on the data. So we gain a lot of data storage, and this is for 100,000 patients, and then we use different number of SNPs. Like whole genome sequencing means you have, I don't know, 50 million, whatever you get from 1,000 genomes, basically. Whole exomes is just the exons, I think a few million. And then the B chip is somewhere around 700K to 1.5 million, I think. And then the ClinVar is basically clinically validated SNPs that if you're only doing molecular testing, for example, to these patients. So the third challenge was the limited and slow functionality. So we can do 4 different queries on this data set when it's still encrypted. One is we can count the number of patients that pass a filter. For example, return me all the patients that has type 2 diabetes and a genotype, a homozygous alt genotype on a known gene. I'm just making it up. And then it can actually filter for those patients and then return it. So it's a vector, basically. The math query is basically you're asking the question, I want to know the allele frequency of this SNP, either in the entire population or only on the patients that has this disease, or only on the patients that has these SNPs. So you can do different kinds of filtering that returns the minor allele frequency of those SNPs, and then you can also compute polygenic risk scores for a given cohort. And then another one which we think that might be very useful for precision medicine applications. You have a patient, you have this patient's genome, you can't diagnose the patients, you don't know what the patient is, what kind of disease they have. You can ask the database, can you return me genetically similar patients and then their phenotypes? And then perhaps there are patients that have similar genotypes and that has certain diagnoses. So all these queries can be done relatively in real time. So as we mentioned, that the homomorphic computing these queries can incur up to 200,000 slowdown. So it's actually not practical if you just implement the vanilla thing. So we came up with certain ideas how we can approximate these queries in a faster way. And at the end, for example, we can do the, I'm guessing this is the count queries for 100,000 patients. So it's 100 second, up to 100 second query time. For the math, it's the same thing. We can actually calculate the polygenic risk scores almost as fast as in plain text, because polygenic risk scores are just multiplications and additions already. You don't need to approximate anything. And then similarity queries are the slowest one. And there's a typo there. It should be 10 to the 5. So it's pretty slow. But it's still, you can still do it in real time. You can a little bit wait and do this. So that's the trade-off that you get with the security, unfortunately.
How is similarity calculated?
You can calculate an L2 norm between two genomes if using it as vector. That's one option. We also give Jacquard similarity between two vectors. So L2 norm is basically very similar to how you calculate the relative search with the King score. So it's very similar to that, basically. How we, yeah.
I have another question about, it was a very interesting idea to find similar patients, and that's a very important function. But, you know, if you don't, there's a lot of ways to say people are similar,
Yeah.
right? That's actually, and so is there a method, like ultimately, I feel like, especially if you're looking at rare disease patients, you're saying, I have this person, they have these really unusual phenotypes, here's their genome, can we find people who are similar? Ultimately, to really use that information, I think you have to go back to the patient record and actually read, and because I have similar functions that I use all the time, and oftentimes you'll find that they're similar in kind of superficial ways that aren't really your intention for that function. And so with this whole process, whole encryption process, do you maintain at some point somebody's ability to actually look at the record and do that kind of query, or is that not feasible in this encryption?
In this particular scenario, it's not feasible, but the goal is that you give an idea to the person if they need that data. So they are able to interact with the data so that they can go through the access process. So that's kind of the idea. And if someone just needs the other queries for their research, all the better. They don't need to go through. But then if you find something interesting, then you can go back to the data owner saying, oh, can we look at this person's records in plain text, for example. So that's the kind of the idea.
So this would be available to anybody who doesn't really have access to the data?
Well, there is still an authorization process, right? You have to send your key to the data owner, and you have to give them some explanations why you need the data.
So I could, in principle, download the PRS of like the 500,000 people in the UK Biobank? With this method?
For UK Biobank, though. So you still have to get permission for UK Biobank, though.
I mean,
Well, if
because
let's say I have.
you decide whether you require more access or not.
Yeah, so the idea, if there was a public-facing querying way of UK, if UK Biobank decided that they are going to do something like this, then you could, in principle, let's say I generated 1 million patients with psychiatric disorders, and you don't know if you want that data or not, I could create a public-facing system like this, and then you send me your public key. I verify that you're a researcher at University of Chicago. I allow you to interact with it, and then later you can say, okay, let's make a data use agreement. Let's exchange data because there are some interesting patients that I want to have access to.
And this is in practice working with some of the private data?
No, it's just the framework. Yeah, it's just the framework. I'm a method developer. I don't generate data, so yes.
I was just curious a little bit about the example of polygenic risk scores that you were studying. So, like, I can see how it would be a straightforward calculation if you were using like a clumping and thresholding type approach.
Yeah.
But it seems like some of the more elaborate modeling approaches for using GWAS summaries that we would.
Yeah, you can't do cross-validation. I mean, at least not in this framework.
Okay, yeah.
But there are papers that does that with homomorphic encryption.
Okay,
Yeah,
okay,
yeah. So yeah, I can send you. There is a preprint actually.
I'll look for you after the talk. Thanks.
Yeah. Okay. So these are what we can do, but do we get accurate results? Because we did a lot of approximation, right? So to show that, what we did is we calculated here I'm showing an example of, I created a type 2 diabetes cohort in UK Biobank, and then I know some of the GWAS variants, and I asked the question in my cohort versus the actual cohort, do I see any difference between allele frequencies? It seems like we calculated very accurately. And then I also did some kind of chi-square testing between case and control, and our p-values were also accurate. We then asked the question, can we actually accurately calculate the polygenic risk scores if I have the effect sizes and which SNPs that are affecting it? Yes, but with an asterisk that because we convert everything to integers, you see that there is a little bit loss of precision. But overall, it's not affecting too much. The distribution is still the same. So, okay, well, I'm running out of time. So the next thing that I wanted to talk about, which I don't know if I'm going to get into it, but there is this, so let's say this is great. You can interact with the genotype data, and you can do basic queries, and now you can keep it very secure. A lot of people are interested in doing machine learning with the data, right? Especially the clinical data. And there is a huge problem because institutions usually don't like to share their data. So in the recent years, Google developed this thing called federated learning, where you can do machine learning across institutions without moving the data between them. So it has also privacy issues because you're sharing gradients, you're sharing weights. It also has a lot of other issues in healthcare, and I wanted to talk about how the applied cryptography can help overcome these issues, and I'm going to talk very quickly. So many of you may know federated learning is model moves, data doesn't move. There is a central server, sets up the model, everybody does local training up to a certain point, sends their weights, it gets aggregated, broadcasted back, and then this happens until it converges. But it breaks down when we have clinical data, because clinical data is often independently identically distributed, right? It's non-IID. There are many ways that it's non-IID. It could be that we are using 2 data sets from 2 hospitals that are specialized in different types of patients. They might be capturing the tests in different frequencies. The missingness might be different. Their diagnostic criteria might be different. In international settings, this happens. And then they might be seeing different patient populations, right? Demographics might be different. And we actually showed that if you have data from Germany versus United States, Colombia, UTH, and Germany, even things that are simple like heart rate can have different distribution. And this doesn't mean that, I don't know why this happened, but this doesn't mean that the distributions, there's a bias. It just means that maybe they are using a different machine, right? So, and this affects your federated learning accuracy because you don't know the data in the others, so you can't assess this. So how do you solve that? There is this new phenomena called personalized federated learning, where you're trying to make either the models more generalizable, or the idea is that you're taking advantage of all the sample size that you have across institutions while also tailoring your model to your own data set. And one technique is called clustered federated learning, where you can cluster the patients in a federated way across institutions and then create a model per cluster so that the similar patients are getting the same model. But there is a problem with that, which is a privacy problem. The existing clustering approaches use aggregated hospital level data. You can't use individual level data because of privacy issues. But when you use aggregated data, you actually will see that the hospital starts clustering per location rather than the characteristics of the patients. So then it leads non-ideal performance. So we then ask the question, can we use cryptography actually to cluster the patients across in a federated way so that you don't actually see the data, but you can still operate on the data? So for that, we use multi-party computation. I'm going to very quickly go over this. Let's say we have 3 hospitals, and each has their own data set, XA, XB, XC. We have ABC. And each of these data sets are linear combination of 3 partitions of the data, A1, A2, A3, B1, B2, B3, and C1, C2, C3. If I have 3 computing parties, let's say I spin up 3 nodes in the cluster, if I do secret sharing, meaning that I share a portion of my data with one computing party, and then another portion with another portion with another. And if the assumption that these computing parties are not colluding with each other, then that means that none of the computing parties or none of the institutions will ever see each other's data. They only see secretly shared portions of the data. And if I do that in a smart way, when I sum them up, I actually get the sum of the original data set. So each party sums their shares and sends it back. And then when the institutions sum them up and divide it by 2, and most of the time we also add some kind of padding noise on it, and then we subtract the noise. At the end, you get the desired results, but you never see other parties' data. So the idea is that can we use this principle to cluster the patients across hospitals and then create federated models per cluster? For that, I'm not going to go into detail, but we use the ICU data. We have thousands of features, so it's a very high dimensional data. So we first run an autoencoder to kind of reduce the dimensionality, and then we did a multi-party computation-based dot product calculation between these vectors of patients using the secret sharing. So you're not sharing the vector, but you're sharing a secret share of it. And then the dot product between patients tells you basically how far these patients away from each other based on the features that we have. And then you can create a similarity matrix using that, and then cluster the patients based on these similarity metrics, and then create a risk prediction per cluster using federated learning. Here, I want to show the results. So the centralized is if you have access to the data in a single place, then that's your gold standards, basically. Single is everybody has their own data, so they don't have quite the sample size. Federated averaging is the vanilla version of federated learning, which is just you combine the weights without doing any clustering. CBFL is the patient level, the hospital level, summary level data clustering. And then PCBFL is our method, where you do the clustering in patient level, but use multi-party computation. So as you can see, it can be almost as good as the centralized model, where you still take advantage of the sample size while you're not biasing your model to the dominant data type. Yes?
What's the computational overhead of doing this?
The MPC is actually cheaper than homomorphic encryption. But the computational overhead comes from the communication cost, because you have to communicate. But in federated learning, you're communicating anyway, so we just take advantage of that.
I mean, the centralized where you have.
Oh, centralized should be very fast, right? Because you.
What's the order of magnitude?
Oh, good question. I don't remember. Yeah, sorry. Any other questions? I'm going so quick because I'm already 4 minutes.
There was a quick one. You relied on some sort of structure
Yeah.
independence, or in your previous slide, you had A1, A2, A3.
Yeah.
Is that something you can achieve just randomly? I guess that's what you recommend, to split the data
Yeah.
randomly?
Yeah. Split the data random, and only I know my splits, so that when I get it back, I can go back to the original one. But moving forward, privacy will become a bigger issue because we keep getting new data modalities. There are more correlations. Even if it's anonymized, you can still link people to different databases. And now what we want to do is actually to connect all the modalities, right? We want to connect EHR data to genetics data to imaging data. So the long-term goal in my lab is to create these privacy-preserving, AI-ready, cloud-ready, user-friendly tools. All of these tools have APIs so that the users don't have to know anything about privacy security. They can just, like for example, interacting with database, they would interact with it as if it's a plain text database. Everything is created in an API. So that's the goal. And with this, I just want to thank my lab and all my funding sources. And thank you so much for listening.